Hi everyone. Hopefully you're all ready for your exam tomorrow. If not, keep studying and come to your professor's review session that'll be happening honestly like a couple hours. Honestly, maybe less than that. But yeah, I just wanted to quickly explain this question again. I know I did. I went over this on Wednesday and Thursday's sessions, but I feel like people were still confused, so I just want to clear up any misconceptions or confusing thoughts before y'all go into the exam. So, these questions has to do with crossing over, specifically single crossing over and double crossing over. Crossing over, as we know, occurs during meiosis. So I have this diagram here. It specifically occurs during meiosis 1, during prophase. Anyway, I want to compare what will happen if no crossing over was to occur versus if crossing over was to occur. So during meiosis, we start off with a diploid cell and we end up with 4 haploid cells. In other words, in this example, we start off with 2 homologous chromosomes and we end up with them being assorted into 4 of these gamete cells. Okay? So if no crossing over was to occur, the chromatids that end up in these cells will look just like the parents. For example, we have our blue chromatids. They look just like the blue of the parent, while the pink looks just like the pink of the parent, meaning no genetic variation or no recombination has occurred. On the other hand, if crossing over was to occur, as you can see here, we have our chiasma, which is where recombination occurs. We get one chromatid and another chromatid that have switched genetic information at that section. So if we go down here and we look at the gametes that were formed, we have 2 of these gametes that have recombinant genotypes. I'll just call them recombinant gametes. We have 2 recombinant gametes. And then, because crossing over only occurs in 2 out of the 4 chromatids in a tetrad, the other 2 will look just like the parents. And this parent. All right. Makes sense? Good. So this question, like I said, has to do with single crossing over, double crossing over. Number 1 is single crossing over, and it's asking what's the probability of having gametes with A to C that have A to C single crossing over gametes. So if we go back and we remember and we look at this map of chromosomes, A is here, C is here. The distance between those two is the percent of gametes that will have that recombination. So if it's asking for the probability of having A to C recombinant gametes, we have to add, because it's giving us 7. So 7 plus 28 is equal to 35. So 35% of the gametes will have a recombinant genotype. That can also be written as 0.35. Either one is acceptable. And now it's asking what's the distance. Like I said, the distance between these genes, they're pretty synonymous with the probability. So the distance would be 35, and the unit is map units. I think there's also like cm, but we'll just keep it as map units, okay? All right, so moving on. If you have a sample of 500 flies, How many progeny are expected to have A to C single crossing over genotype? All right. So we're looking at the same recombination. So we can just keep the 0.35 and multiply that by the number that was given, so 500 flies, and that is how we get our expected number of recombinant progeny or recombinant gametes. And so just multiply that and you'll get your answer. All right, so what's the probability of having an A wild type, B wild type, C wild type gamete? Now, if we look closely at these genes, or if we look closely at these chromosomes, we can see that this chromosome has a genotype that is A, B, and C, while this chromosome has A wild type, B wild type, C wild type. The fact that this one is different from this one shows that these two have recombined. The C's of those two have recombined. Okay? And it's asking for the probability of having this genotype specifically. So if we remember, after meiosis, we get 4 gametes. 2 will be the parents, so I'm going to put a P. 2 will be recombinant, so I'm just going to put an R. We're looking at the distance between B and C, and that's 28. So the probability of having a recombination from this portion to this portion, which is basically 28, is, 28. 28%. However, it's asking for this genotype specifically. So we have A plus B plus C, and then we also get A, B, C plus. When I say plus, I mean wild type. Please don't confuse that with something else. But anyway, we have to divide this probability by 2 because all of them won't have this. Half of them will most likely be this too. So we have to divide 28 by 2 and we get 14. So 14% or 0.14 will have this genotype. Does that make sense? Okay, cool. All right. Lastly, number 4, what's the probability of having double crossing over progeny in a sample of 500 flies? So we have to use the product rule to solve this. And we use the probability from A to B, which is 0.07, and then B to C, which is 0.28, and we multiply them to get your probability, and then you can multiply that probability that you get by 500. So if you want to bypass all that, you just multiply these 3 numbers together, and that is, whatever you get is the number of double crossed over progeny in a sample of 500 flies. Hopefully that made sense. Please send me any questions. Come to your professor's review session, like I said, if you have any more questions. Good luck studying. Good luck on your test tomorrow. Yeah. No